Welcome, everyone, and thank you for joining us for today's webinar, Better Knowledge, Better Care, Precision Medicine for Any Cancer, Any Assay on Your Platform, presented by Dr. Sheryl Elkin, the Chief Scientific Officer at N of One, a QIAGEN company, and Dr. Dan Richards, Vice President, Biomedical Informatics at QIAGEN. My name is Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit them at www.qiagen.com. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen, type those questions into the drop-down box. Our speakers will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, please click on the Ask a Question box and let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. Now, without further ado, please join me in welcoming Dr. Sheryl Elkin and Dr. Dan Richards. I will now turn the presentation over to them. Welcome.
Thank you. This is Dan Richards. What I will talk about first to start us off here is an overview of QIAGEN Clinical Insight, which is a platform QIAGEN provides for the interpretation of variants from next-generation sequencing, both for cancer and other hereditary diseases. Our focus today will be on somatic cancer testing and interpretation. And with that, let me give a brief overview of how this QIAGEN Clinical Insight fits into the overall workflow, laboratory testing for hereditary or somatic cancer. So basically, samples come in to the lab. There's a test menu that's supported by QCI Interpret. Somatic cancer, myeloid neoplasms, as well as hereditary cancer carrier screening and other applications are supported. You can use any sequencer to perform your sequencing, although QCI also provides an optional secondary pipeline, analysis pipeline called QCI Analyze for translating your raw sequencing data into detected variants that you can use whichever sequencing analysis pipeline you prefer. And QCI Interpret takes a test product configuration, which is a set of genes or biomarkers, copy number, fusions, whatever you may be looking for in your particular assay, as the input, along with patient case metadata, for example, to enable specific clinical trial matching, so information in a case such as the patient's age and gender, demographic information beyond their diagnosis, for example, as inputs. And then it applies the content that I'll talk about briefly in terms of information from the clinical literature, practice guidelines, and other sources to put those alterations that are detected by sequencing into the context of the cancer they are detected within to produce a final report that can be signed and returned from your lab to physicians that are ordering it. And we also provide a reporting policy that streamlines that process or automates it to a large degree based on the AMP guidelines for interpretation and reporting of somatic variants. So some of the challenges that are faced in doing this interpretation of the alterations that are detected in tumors and putting them into context and deciding which ones to report out are twofold. One is determining the biological relevance, so whether they drive tumor progression or not. So that in the standard terminology is the notion of pathogenicity. It's trying to ascertain which ones are activating mutations in oncogenes, for example, or loss of function mutations in tumor suppressors that may be tested on the somatic cancer panel. And there's a constantly growing number of publications that characterize the role of different alterations in tumorigenesis, functional studies and case reports, for example, as well as frequency information from various sources, both to help identify which variants are common in the general population and the germline and can be removed for those reasons, as well as clinical case reports of the variant in tumors from patients. The second aspect is clinical relevance, or the actionability. So not all pathogenic alterations in tumors can be treated currently. The actionability is specific to the diagnosis of the patient. So there may be things that can be done for non-small cell lung cancer patients, alterations that are not on indication, for example, for a patient with a rare type of cancer. But actionability is considered in terms of what therapeutic options a patient may have, clinical trials that may be available for new treatments, but also prognostic significance of various alterations in terms of the outlook for their ability to survive the cancer, and diagnostic significance in some cases for blood cancers, for instance. And so keeping up with all the changes in drug labels and the trials and the constantly growing number of clinical studies makes comprehensive and up-to-date reporting, both for pathogenicity and actionability, a challenge. And that's one of the things that QIAGEN addresses with products such as QIAGEN Clinical Insight, and as we'll talk about later in this presentation, N of 1's offering of for interpretation as well. So every month there's several new cancer drugs approved. There's hundreds of clinical trials that open, and then there's typically hundreds of trials that stop recruiting, whether either temporarily or due to completing the trial across the many sites that offer these. And there's every year over year, there's a constantly growing number of publications on somatic cancer. So all these are some of those challenges I referred to in terms of current knowledge that's needed to bring to bear to interpret somatic variants. So for the clinical testing lab, there's a couple challenges. You need consistent, reproducible interpretation across the team providing the report product. You need immediate access to current information about actionability of the alterations. You need transparency or visibility of the underlying evidence to support the treatment options and support what's reported out in your final reports. And there's a need for simplicity and fast turnaround time, as well as guideline compliance. And so that's one of our focuses at QIAGEN, is really helping ensure high quality and consistent reporting across all the labs performing interpretation using our products. So based on the guidelines, there are guidelines from AMP and ACMG for germline testing. There's also that pertain primarily to pathogenicity. That's assessing whether the variant drives disease progression. And so variants are put in the categories from pathogenic to benign, one of these 5 categories each. On the actionability side, there are tiers, according to the AMP guidelines for somatic testing, that indicate to what degree a particular alteration or variant biomarker indicates a particular treatment option. And so there's strong clinical evidence tiers, there's potential clinical significance, and then there's similar to variants of uncertain significance, or VUS variants in the pathogenicity context, there are tier 3 or unknown clinical significant variants in the actionability context, and likewise tier 4 correspond to likely benign and benign variants. So QIAGEN Clinical Insight is a product that's delivered on the internet. It's secure. It takes as input the alterations detected by next-generation sequencing and puts them into biological context. It supports many different indications, both rare and hereditary diseases, hereditary cancer, but what we'll focus on today is somatic cancer oncology testing, both for blood tumors, myeloid neoplasms, as well as solid tumors are supported. So briefly, what is QIAGEN Clinical Insight? It's a web-delivered application. You can upload your the variants detected in each sample along with the metadata for each case to the system through APIs, or if you're lower volume, through the user interface. It lets you see each alteration in the context of the current evidence that QIAGEN's curated for you, and through computational means puts those alterations into context. So it's what we refer to this as is evidence-based clinical decision support software. It assists you in the classification and reporting of alterations, independent of which next-generation sequencing platform you use. It provides comprehensive annotation and literature resources, so it displays the information about the alteration in terms of its relevance in a particular cancer type, reported clinical cases in the cancer type, prognostic information about outcome for patients with that alteration when it exists in literature that we've curated, as well as treatment options and specifically matched clinical trial options. It's fast and automated, so we apply the AMP guidelines and have a means to apply that in an automated way to give you a draft report from which then you can review and refine if you see fit to get to your final report in a streamlined way. And it provides us treatment and trial identification. So we keep up to date with the latest NCCN guidelines, all the drug labels, as indications are expanded and new drugs are approved. All those things are curated and available and automatically matched to alterations you detect so they can be included on your reports. And we also provide customized reporting as part of QCI. Many of our customers use QCI to generate their final signed reports. There's a state mechanism, so you can identify which reports are ready for sign-out, do the review, and do the electronic signature. We also, through our services, provide customization of the report, so you can have essentially the report look any way you like it, and we can help put the relevant content automatically into the report template for your particular test product. So this is all powered by QIAGEN's knowledge base, which enables QIAGEN Clinical Insight, and it standardizes the high-quality clinical evidence that we curate from the biomedical literature in terms of primary evidence from figures and tables and text supplementary information from articles, as well as from other sources such as practice guidelines and drug labels. QIAGEN's approach is quite unique in terms of we capture the information not just as typed in sentences, for example, with QIAGEN Clinical Insight, but in a structured manner. That structured manner enables the evidence to be written out, so to speak, automatically in templated English, like this example, what we call a finding, a natural language generation from the structured evidence we captured into a knowledge base, but it also enables it to be used computationally. And so that the power of the computation is that when you provide a variant to the system can automatically take that structured knowledge and compute the ACMG classification, dynamically compute the current AMP classification without somebody having to manually update each of those all the time, since there's many hundreds and hundreds of thousands of variants and constant change in terms of new evidence and new implications for treatment and trial matching. This enables us to cover a very broad breadth of information about alterations and their clinical significance and clinical actionability in a very streamlined, automated, and high-quality, consistent way. And so aside from the QIAGEN knowledge base, where we curate from the primary literature and guidelines in our streamlined way, we also integrate other sources, external databases, and clinical trial sources like clinicaltrials.gov. We curate as well as drug labels to create a comprehensive view of relevant evidence for interpretation of somatic variants. And the knowledge base is updated weekly. So there's new clinical trials open and close on a weekly basis. Those are reflected in drug labels, indication expansions and approvals. New primary literature is constantly being published and that we curate, as well as the roughly quarterly, depending on the particular cancer type, drug guideline update. We keep those up to date as well. And so all that information sits in QIAGEN Clinical Insight, and when you send it the particular alterations you detect in your tumor sample, it immediately applies that current knowledge in the framework of those guidelines to put those alterations in the context and produce a preliminary report. So the product itself, once you look at a particular case, you can sort of see a list of the cases in the system that are pending sign-out or pending a review. When you open a case, you see a user interface along these lines. It has a tile for each alteration that's detected in the tumor. It supports sort of the wide variety of alteration types, including single nucleotide variants, indels, copy number changes, fusions, those sort of things, as well as things like TMB status and MSI status can be provided to the system as well. And each alteration is put into this computed classification, both for actionability in terms of which tier in the particular diagnosis for that tumor is appropriate based on available treatments and trials and other evidence, as well as the pathogenicity classification based on whether the variant drives tumor progression. So for any variant, you can click on it to see the evidence supporting its classification. And so in the AMP context for actionability within the diagnosis, there's a wide variety of criteria in the AMP guidelines for somatic interpretation. It shows you which of those criteria are met. There's in the user interface, there's little links to the evidence, which you can click on to get more information. In the primary source material that we curated the evidence from. You can also get a summary of the particular criteria, both for actionability and for pathogenicity that come from the guidelines. These, those acronyms at the beginning, the 1A is Tier 1A, the PA1 is one of the about 30 or so criteria in the guidelines for pathogenicity classification, for example. And these just let you see at a glance the lines of evidence that support the classification of actionability and pathogenicity, which enables a quick review for variants that you may be seeing for the first time, which is quite common. As I mentioned, you can click on the evidence for any criteria to see the supporting sources. So that brings you directly to the literature cited that we curated the evidence from, and it also shows those sentences rendered out from the structured computable knowledge that we curated that support the particular line of evidence of the particular criteria, and shows you sort of a summary of the primary evidence that was curated. We also provide this ability to customize your report. So we provide a sort of a default report template. It has the general information about the patient, the specimen, and the client that ordered the case. Lets you provide an overall report comment if you so choose. Summarizes what was detected in terms of positive results for the case. And then provides a table of particular alterations that were detected, information such as allele fraction, and the which therapies were approved for the patient's cancer type, as well as are available for other cancer types for reference in clinical trial, top clinical trials that are most suited for the patients that take into account both eligibility and ineligibility criteria in determining those top trials. Follow-on pages have information about the particular clinical trials, locations, and contact information, things like summaries of the alterations and additional supporting evidence for the assessments that are made in determining the final report. Again, these templates are customizable, and so if you launch a new somatic cancer panel, the QIAGEN services team would be happy to help customize the look of the report you could generate off QCI to your particular laboratory's satisfaction. QCI has been used for many hundreds of thousands of samples by laboratories worldwide. QCI is available in most of the world currently. And it's gone through several independent validation studies by leading sequencing laboratories that sort of compared their prior sort of fully manual approach to the streamlined automated approach that QCI provides. And we have extremely high, from those analyses, those labs found very high concordance in determining which variants to report and the relevant content both in somatic cancer and hereditary cancer testing, as well as a significant time saving. So QCI, since it provides all this evidence, current evidence and context, it very greatly streamlines the time it takes to produce a report, review it, and sign it out relative to other means of sort of manually searching for some of that evidence or trying to collect it oneself in an independent way. So overall, QCI interprets, standardizes next-generation sequence interpretation and reporting. It automates the guidelines, which provides a scalable, standardized, and reproducible means to do interpretation and reporting of these type of tests. It requires very little upfront effort, since essentially all this information is pre-curated. It's all at hand, so you can readily adopt the guidelines, as well as through QCI have access to all this current information to produce actionable clinical reports in a very efficient way. With confidence, too, because we provide full transparency to the supporting clinical evidence, and we have over 20 years experience curating primary evidence from the clinical literature with a high degree of accuracy and high degree of comprehensiveness. And so that standardized clinical decision support and reporting for any next-generation sequencing assay, sequencer, platform, or pipeline to produce those variants. So we recently announced the acquisition of N of 1. This is one of these sort of the leading company in providing manual review of somatic variants. And it's a complement to what we provide with QIAGEN Clinical Insight, which lets you have quite a degree of flexibility in terms of deciding which variants to report and refining the clinical trials, for example, and doing things in a semi-automated way. N of 1 provides sort of a final refined product directly, and Sheryl will talk about that next. But overall, the QIAGEN provides the knowledge that powers actionable insights for both germline and somatic cancer. And then we have a number of solutions here. QCI Interpret, which I just provided an overview of, lets you do the analysis of variants and interpret them, produce a final report. There's other products like HDMD and genetic variant analysis that assist in that process too. For secondary analysis, if you're developing a new pipeline that you want to validate, we recommend considering QCI Analyze. There's also the Biomedical Genomics Workbench that QIAGEN provides to take that raw sequencing data and translate it into variant calls, copy number calls, fusion calls, for example. And next, I'll transition us over to Sheryl, who will talk about N of 1's offering, which is an on-demand curation and clinical interpretation service that we offer. So with that, Sheryl, it's all yours.
Thank you very much, Dan, for the terrific description of QCI and a great lead-in to N of 1. As Dan mentioned, N of 1 is now part of QIAGEN, bringing our clinical interpretation solutions to complement QCI solutions. So who is N of 1? N of 1 has been in business for over 10 years, and we have grown and evolved with the field of precision medicine. We were founded in 2008 when we began working directly with patients and their physicians to help them identify the most appropriate molecular tests for their tumors, actually help them to get the test done, and then explain the results so that the doctors would have a way to use those results to help their patients. And through the process of working with individual patients, N of 1 began to establish a knowledge base of biomarkers, of genes, of variants in the context of different cancer types, linked with the drugs that had either emerging or established evidence that they could target those biomarkers. By 2012, we had begun working with large hospital systems and commercial laboratories, taking in the results of patient molecular tests and providing reports back that the hospitals and laboratories could then sign out. And by 2016, we'd partnered with over 30 clinical laboratories to provide this clinical interpretation, which brings us to 2019 when N of 1 joined QIAGEN, having generated reports for over 100,000 patient cases and having established a deep knowledge base that covers over 1,800 genes and over 100,000 unique variants that have been curated and analyzed. So what value does N of 1 bring to our partners? Why do they like us? Our reports are prepared by an experienced team of PhD scientists that are supported by a team of practicing oncologists. So our oncologists review all of the clinical data that we collect and analyze and describe for clinical validity. We rely on people, process, and technology. We have a sophisticated proprietary knowledge base with a set of proprietary curation tools that help our scientists to curate the literature efficiently and accurately. And we utilize a method of knowledge engineering that facilitates efficient and accurate report generation so that information can be stored in one place and used in many places. Our process, both our curation and our software development, is controlled by a quality management system, ensuring both high quality and reproducibility. Our product is highly scalable because of both of our knowledge engineering and the economies of scale derived from working with many clients. We are able to return reports quickly and expedite the sign-out process for our clients, who can simply review the reports and sign them out. They don't have to go. We provide all the references so they can check whatever they want to check, but they are not required to do their own work to read all of the literature and pull together what are the most important pieces of evidence. We integrate securely and seamlessly with our clients using either SFTP or web services, and we integrate with any bioinformatics solution, including QCI. So looking at the over 100,000 cases that N of 1 has reported on over the last 8 years, why is clinical interpretation important? Why do you need something like N of 1? Over the last 20 years, the number of different treatment options for patients has grown exponentially, whereas 20 years ago you might think of HER2 and trastuzumab in breast cancer, maybe EGFR was starting to emerge as a biomarker in lung cancer. Those numbers have increased dramatically over the last 20 years. However, only a small percentage of cancer cases actually qualify for an FDA-approved therapy based on a biomarker. And when you look at a distribution curve looking from on the far left at the percentage of cases where there is an FDA-approved drug that's predicted, where responses could be predicted by a biomarker, all the way to the far right where there's no targeted therapy available, it turns out that only 16% have an FDA-approved therapy that's the most applicable option. And this number could even be an overestimate because in most types of cancer, because non-small cell lung cancer, about 20 to 25% of cases have targeted therapy options that are covered under the guidelines, and so that draws the average up. In 21% of cases, there were no targeted therapies that were appropriate for a patient, but in the center of the curve, 63% of cases could qualify for a clinical trial or off-label use of a therapy. And these are the cases with the most critical need for clinical interpretation because identification of the variant alone is not sufficient to support a clinical decision. The physician needs to know whether that specific variant is likely to predict sensitivity to a therapy. What is the evidence that that variant is actionable? What is the evidence to support the use of that therapy in that disease type? And which clinical trials are available that the patient can access. And this is the solution. This is the problem that N of 1 addresses. So where does N of 1 fit in this workflow? The maroon box labeled clinical interpretation shows the N of 1 position. N of 1 is not a lab. We do not do any sequencing. We don't do sequence alignment, variant calling, or filtering. The bioinformatics pieces you can do using QCI. N of 1 starts at the point after the variants have been called and filtered. N of 1 can receive a filtered list of variants from any bioinformatics partner, and then provide annotation of the variants with evidence for their activity, association with therapies, prognostic or diagnostic relevance. And that information is assembled based on reporting logic that is included within our platform. We generate a report and send to the pathologist for the review and sign out. So N of 1 can report on any type of cancer, solid tumors, hematologic malignancies, lymphoma, and from tests derived from any platform or tissue source, FFPE, ctDNA, et cetera. The reports are specific to the cancer type and generated on a per-patient basis. We provide links between the biomarkers and targeted therapies or immunotherapies. We provide prognostic and diagnostic information, particularly for hematologic disorders, and always provided with the relevant scientific and clinical evidence so that the information we provide is very transparent. You can see exactly where it came from. All of the references are provided, along with a summary of the information. The reports also include a multivariant analysis. So if there's a combination of variants either within a gene or between different genes that results in a different drug sensitivity or resistance than for any of the individual variants alone, that summary of that information will be provided and it'll be highlighted in the report. Any effects of multivariant analysis on prognosis or diagnosis are also included, again, very important in hematologic disorders. The reports also include clinical trial matching. The clinical trials are provided for each variant in the report. They are filtered by molecular eligibility, by age and gender, and prioritized separately for clinical specificity and geographic specificity. There are some patients that are unable to travel and need to enroll in a trial that's close to their home or in their physician's own institution. There are other patients that may have a rare biomarker and they would like to seek out a trial that may not be geographically co-located, and so we provide both of those types of prioritization. Most of the tests that we interpret are next-gen sequencing tests, but we can also bring in results from other types of testing from platforms, from FISH, immunohistochemistry, copy number analysis, cytogenetics, and we can integrate the results from all of those different tests into a single report. So again, in heme disorders where you may have cytogenetic results, a send-out FLT3, and a next-gen sequencing test, those can all be integrated into a single report so that the implications of all of those tests can be weighed in the report. So N of 1 offers two different reporting solutions off of our same database and really giving the same type of report. One is fully automated and one is semi-automated. And again, they're drawn from the same knowledge base using the same reporting logic. So either the Rapid Insights or the Precision Insights reports, both are of the same high quality. The Rapid Insights report is fully automated. They can be turned around in seconds to minutes. With a full report. If a variant hasn't been previously curated within our knowledge base, it's returned as variant not curated, so that there's no question between a variant that is unknown in the literature versus a variant that the N of 1 scientists simply haven't analyzed before. However, those reports, if a client receives a report with a variant not curated, it can be reflexed to N of 1 for full curation. And N of 1 does keep track of the variants not curated that come in. If any variant comes in more than twice, it is placed into a queue for curation so that the next time it comes in, it goes out automatically. The precision reports are semi-automated, so any information that's up to date in our system and meets all of our time and quality standards would be automatically assembled, and any new variants or new genes and disease contexts would be curated on demand. And the turnaround time for the Precision Insights reports varies by panel size from hours to up to 3 days maximum. The average for all of those reports is less than a day. And for those reports, every variant would be curated. So the reporting that we do, similar to the guidelines used by QCI, we report using the guidelines described in Lee et al. In 2017, the AMP ASCO CAP guidelines. It's a tier-based reporting system with levels of evidence that are defined in that paper. And to give you a sense of our workflow, a test may be performed, the results are combined with case information, a diagnosis code, an age, a gender, location by state or physician zip code, and compiled into an N-of-1 report request XML that's standard across all of our clients. And that's transmitted into the N-of-1 system using either secure web services or SFTP. Our process is entirely HIPAA compliant, but we don't actually exchange any PHI. It's not necessary to the reports, and we don't generally pull that in at all. And then our reports are delivered either as a PDF or in most cases as an XML. And for the XML, all of the information we're providing is in discrete data elements that allow our clients to design or assemble their own report. And the N-of-1 implementation team assists with implementation and mapping of those data elements from our XML into the client report. And we do provide our PDF to those clients as well for validation purposes. And for clients that are not ready to produce their own report or they don't wish to differentiate using our report, we do produce a PDF that can also be used. So I'd like to give a couple of examples of what the reports can show and what they actually look like. And this first example is a case of a 51-year-old woman with non-small cell lung cancer. And as you can see on the report, it's divided into tiers, and we have tier 1, tier 2. In this case, there were no tier 1 mutations and there were no variants of unknown significance. The tier 1 mutations would be mutations with strong clinical significance, therapies that are approved by the FDA for that alteration. There were none of those in this case, but there were 3 tier 2 alterations in NRAS, CDKN2A, and RB1. And for each alteration, we show the gene, the specific variant that was identified, therapies that would be approved to target this alteration, this specific alteration in this indication, therapies that have been approved in other indications, along with the level of evidence in parentheses, and whether or not any trials are available. If there were any prognostic or diagnostic variants in this case that were relevant in this specific disease, they would also be shown in a separate table. Guidelines would also be shown on the front page. If there were guidelines specific for this cancer type, they would be shown, but the guidelines are only shown if they're relevant within that cancer type. So in this case, an NRAS variant would have guidelines that are relevant in colon cancer that would be summarized, but for non-small cell lung cancer, there are no relevant guidelines. This report also highlights the multivariant analysis showing the interaction. CDKN2A is under investigation as a predictive biomarker for CDK4/6 inhibitors, but requires wild-type RB1 for the mechanism of action. And there's also an RB1 mutation in this report. And so the interaction summarizes that information and provides references for that data. There were no variants of unknown significance or benign variants. Those would be tier 3 or tier 4, so they're not shown here. And so as you move deeper into the report, we show more and more specific information. And so I'll walk quickly through one variant from this report to give you an idea of what the information looks like. In this case, it's an activating NRAS mutation. And the first section, the biomarker results summary, is intended to give the take-home message. What kind of a mutation is it? What does it do to the gene? What effects does that have biologically? Are there any drugs available to target that alteration, or does it have any prognostic or diagnostic significance? And that's summarized in a couple of brief sentences. So if the physician wanted to read only one piece of information, they could read the biomarker results summary and get the information they needed. But each section that follows gives a little bit more, so that if a physician is using a report to support tumor board analysis, or if they're unfamiliar with this mutation and we've determined it to be an activating mutation, they can read the molecular function section and understand exactly what is the evidence that supports that information. So the biological relevance is divided into molecular function and incidence. The molecular function describes what does this variant do, what is the evidence. It provides references for all of that. Q61L in NRAS disrupts GTP hydrolysis. results in constitutive activation of the mutant protein, and that's all supported by references as well. Incidence of this gene in this disease type, the data is derived from COSMIC, from TCGA as cataloged in the CBio portal, and also from the scientific literature. The next sections get more into the clinical relevance of the alteration in this specific disease type. So looking at role in disease, this section provides information for cancer in general and also for this disease specifically. And if there's very specific information about the specific variant, it would be included here as well. Describing what role does this gene play in cancer. It activates pathways downstream in lung adenocarcinoma. It may be exclusive of other drivers, et cetera. And then the next section looks at the effects that this mutation or other mutations in this gene could have on drug sensitivity and drug resistance. And it could be downstream effects, looking at MEK inhibitors or MAP kinase inhibitors to target NRAS, which can't be targeted directly. We would also look at the effect that this gene could have on resistance to other drugs. And then getting even deeper into the information, we provide clinical evidence, that is, summaries of specific data from clinical trials that supports the summary information that came earlier. This information is specific to this particular disease, so in this case, specific to non-small cell lung cancer, and organized by the phase of the clinical trial from which the data is derived. We'll present the highest level data for any given drug. We would not show phase 1 data for a drug for which we're presenting phase 2 data. We only show preclinical data if it's very specific for the disease or the variant, or for a new or novel therapy that has already advanced into the clinic. We provide a list of sample therapies, the name, the brand name if it's been approved, the level of evidence according to AMP, and the target or rationale for use, as well as the highest level of clinical development. How far has it advanced through clinical trials? And then, as I mentioned earlier, the clinical trials are presented two lists of 5 trials for each biomarker. The trials come primarily from clinicaltrials.gov, but can also come from a few private sources. And the trials are matched based on either a single biomarker or multiple biomarkers. So the filtering of trials would take into account if there were PIK3CA mutation and it said may not be used, the trial is not eligible, or the patient is not eligible if they have a KRAS mutation, that trial would never be shown for a patient with a KRAS mutation. And the trials can be prioritized by institution. They're filtered for age, gender, disease, and then, again, subjected to the different prioritization rules by clinical specificity in terms of specificity for the disease, for the particular variant or a particular type of variant within that gene, and then separately for the geographic specificity. Now, I'd like to walk through a couple of other cases just to highlight a couple of additional features of the N of 1 reports. And one of the key components that comes from N of 1 is the very specific variant classification and description that's done from reading the literature and evaluating all of that data. Detailed and accurate variant characterization is critical for physicians to be able to make the best treatment choices for their patients. So in the first example, we're looking at a series of reports for non-small cell lung cancer with EGFR mutations. And in this first case, the patient has the most common sensitizing EGFR mutation, which is L858R, level A evidence for using a number of different therapies. You can see the guidelines are cited because L858R is specifically mentioned in the guidelines in non-small cell lung cancer. And that was a tier 1 variant. However, not all EGFR mutations are the same. In the second case, we have a non-small cell lung cancer with an unusual E330K variant. So most physicians would never have seen a patient with this mutation. It's not on the label for EGFR targeted therapies. However, biochemical studies have shown that this is an activating mutation. And in this case, the variant is displayed in tier 2. The therapies have either level C or level D evidence, and they would be available either off-label or through a clinical trial. And although the EGFR-targeted therapies are not specifically approved for this mutation, they may still be a good option for the patient. And the specific information supporting both the biochemical description of the mutation and any clinical evidence that's available would be provided to the doctor. In contrast, another uncommon EGFR mutation, R1031L, could be detected. And this is a variant of unknown significance. It's never been reported before. It hasn't been functionally characterized. And for this variant, EGFR-targeted therapy is likely not the best choice because it's unknown whether this variant would predict sensitivity or whether it's simply a polymorphism. So the N of 1 report is providing the evidence to distinguish between the Tier 2 E330K, where EGFR inhibitors could be a good option, and the Tier 3 VUS R1031L, where a different possibly chemotherapy or immunotherapy would be might be a better approach. But it's providing all of that information so that the physician can weigh it and make that decision. In another example, we have an FGFR3 S249C mutation in bladder urothelial cancer. And in this case, we're showing how N of 1 can provide clinical evidence to support the use of therapies that may not be approved in a particular context. So FGFR3 S249C is a well-known activating variant. Several tyrosine kinase inhibitors include FGFR3 among their targets and have varying levels of evidence to support their efficacy as an FGFR3 targeted therapy. So the clinical evidence presented describes the efficacy of the therapies in a specific clinical setting as they have been tested. So this is a bladder urothelial cancer case, and phase 2 data is presented for drugs studied in urothelial cancers, drugs that have been studied in the context of FGFR mutations in solid tumors, as well as drugs studied in the context of FGFR mutations specifically in urothelial cancers. So a physician can weigh that information and decide whether they think that a trial of a kinase inhibitor that targets FGFR3 might be the right decision for their patient. And the available drugs are shown in a table ordered by their level of evidence. And then finally, the N of 1 reports can also highlight rare but highly targeted mutations with drugs that may only be accessible through a clinical trial. So before the approval of larotrectinib, NTRK fusions had been identified as clear drivers in certain cancers, and entrectinib and larotrectinib were available only in clinical trials. So the drug table shows the level of evidence for the available therapies and distinguishes between therapies that have a higher level of evidence versus a lower level of evidence. So crizotinib is a drug that's been approved by the FDA and other molecular indications for ALK and ROS fusions, but only has level D evidence for NTRK fusions. It hasn't been approved for NTRK fusions in any indication, although biochemically it's been shown that it could potentially target NTRK fusions. However, the then investigational drugs, larotrectinib and entrectinib, were in active clinical trials specifically for NTRK fusions, giving them a level C evidence according to the guidelines described by AMP. So the data in the report supported the selection of a clinical trial of an investigational therapy, potentially over an off-label alternative. And the trials are then presented to the patients, or the trials are then presented in the report so that the doctor and the patient can consider whether that may be the best option. And in the XML version of the report, there's more detail on each of the sites so that a trial such as this one, which has many potential sites, the patient may be able to find one that's nearby. So to summarize, N of 1 can transform molecular results into actionable insights, supporting physicians' decision-making at the point of care. And N of 1 joins the family of QIAGEN products along with QIAGEN Clinical Insight, HGMD, and Ingenuity. And together, these products power actionable insights for both germline and somatic sequencing data. These products are highly complementary. QCI supporting in-house curation, taking advantage of a vast curated database to drive computation of pathogenicity and actionability. N of 1 picking up providing comprehensive variant interpretation, report writing, integration of results from multiple assays in a scalable platform. The QIAGEN bioinformatics solutions allow the user to proceed from a FASTA file to a clinical report using best-in-class components for secondary analysis using QCI Analyze and the Genomics Workbench, variant analysis and classification using QCI Interpret, HGMD, and Ingenuity variant analysis, and deep, comprehensive, on-demand clinical interpretation from N of 1. The QIAGEN products can truly take the process from sample to insight, from DNA extraction, library prep, and enrichment, to sequencing, to bioinformatics, secondary analysis, variant interpretation, and clinical reporting, providing a single solution from end to end. And with that, we would be happy to take your questions. Thank you very much.
And thank you, Dr. Elkin and Dr. Richards, for that informative presentation. I'd also like to thank QIAGEN for making today's educational webcast possible. As a final reminder, Dr. Elkin and Dr. Richards will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We will see you next time. Bye-bye.